So the plan is to continue north? Plan is to continue north until we're on a contour
We're
to
just
head to the waypoint?
so we're just about to finish that north move to match the contour that the waypoint's on, and the waypoint is to the east, northeast. So once we reach that contour, we'll kind of strafe along it to the east. That sounds all right.
That sounds great. Is that something that we could run a transect as we made that move?
Gonna attempt it, sure.
What's that?
It's gonna depend on the, you know, just as before, when we're doing that and moving laterally in these canyons, there'll be some starts and stops, but we'll try to keep it as consistent as we can.
Yeah.
For a transect.
So we're thinking about starting a transect then?
Sounds like it.
All right, so whenever we get to that contour.
Yeah. It'll come up a bit. About that.
That'll cross over shift change though, won't it?
Yeah, well, so if you want to wait just a couple minutes, we can pass that on and have them start it.
Okay, that's probably a better idea than to
I think so.
Good.
Once they get comfortable.
have a big pause in the middle of it.
Yep.
We can keep transect going through shift change.
Yeah, haven't had too good of luck with that.
Really?
Yeah. They've had some pauses.
Yeah, it's
only
a
in
deal.
less than 20 minutes away.
Uh-oh.
Right.
Lost it.
Yep, okay. It's red, so.
Take over that thing.
Stand by, everybody. We're just dealing with something.
Now I'm forgetting.
You got it?
I can't get to mine. There it is.
Yeah. Move him out of the way and restart.
Not that one.
That's the one. One
Which
lower.
This
lower. Yeah, I believe that's it.
Got it.
Got it?
Got it.
That's not frustrating.
Ooh, that was a...
Nice, Michael.
Yeah, good job,
save.
Man. I'm over here
I
saving
think
everybody.
I think I need a mouse.
Yeah, set it up a little better over here if you don't mind, Michael. Just organize that. All right, let me mark that again. I'll put that in the observations. It's going into engineering notes.
I wonder why they put that box away.
Which one? Sorry.
They got.
I bet if I would guess correctly, it's that it annoyed Bob to have that something moving in the bottom.
So while that we know the red light comes on.
Yeah.
When the red light's on, the vehicle's on a holiday.
Yeah, it's happening enough times that needs to be addressed.
I think. That happens on a launch, it could be fatal.
Yeah.
That was the top, right?
Yeah, lost topside.
There was a crash? Oh, topside crash. There was another crash.
Topside holiday.
When was that?
That was like at 5 in the morning, 5:30.
Really?
Yeah.
Ah.
Well, that's
Frequent.
It's a nail in the coffin for the Frankennuck.
Yeah.
Another nail.
We'll talk to Tim.
Some Primnoa, that's probably Peristanella.
Probably which Primnoa?
Hey, Miley, you can zoom there. I'm too close, but
yeah,
Parsinella.
Hold on, I will type.
I was gonna ask. About a crash that happened earlier, but it's in the log, but it doesn't say anything else.
Thank you, Erica.
It just says crash, so I didn't know if they bumped into something.
As in crash bang?
Yeah, that's what I thought. And then I read the defects when I was writing it in. I was like, oh no, it was a
Thanks for
crash.
the zooms, Marley.
Zoom. We're good?
Yeah? asking, Alex asking about a zoom on the tubes. Are we talking about the red ones that we were seeing earlier? And yeah, is that sea star in the sponge? It looks like that sea star is just fully hanging out in that sponge.
I think it is. I think the comments are about the geology, so the consistency and texture of the kind of cliffs we've been looking at.
Oh yes, the tube-like.
Yeah, should we take a look at that?
If we see it again, have we? I saw it back in that steep bit in the box canyon.
Yeah.
Okay, well, if we pass it again, we'll take a look.
Yeah.
So, is that moving on? Is that
Yep.
what I heard?
Yep.
Okay.
Yep.
Zoom out, thanks. Another
Yeah.
victim over here. Oh, they're everywhere.
Put Argus into that little canyon there if you want.
Sure.
Hey, Michael, you're on the wrong screen
Oh,
now.
dang. I knew that. I knew that.
We'll switch it back before you do it. All right, so
I was like, why does it look different?
yeah. You can switch Dan back over here.
Oh, yeah.
Huh?
Yeah,
No, you're good.
he's looking at Arguses.
Yeah.
There you go, that's good. Okay, back to it. 015, how about that?
Sure.
Bridge now.
That's not one of the sponge we want to photo mosaic,
That
is it?
it is one of the those,
stand
but
by.
I think we're looking for more of a cluster if we
Oh,
can
right.
find that.
Okay, 20 meters, bearing 015. Thank you.
Big sheepdog.
Whoa.
I did not check the winch this morning. Forgot all about it.
Oh, this big
Wow.
cluster.
Interesting angle that they're growing at.
Yeah.
Small ridge with some pretty big primnoids.
Lined up with the current maximum feeding efficiency.
Yeah.
What's that?
They're fanned out to take advantage of the current.
Oh, yeah.
That looks kind of like a chrysogorgid.
The one on the left?
Yeah,
the
does
orange
anyone
one?
want to back me up on that?
It
Am I making that up?
does look like a chrysogorgid.
Like a,
Can't tell.
I don't know though.
Bring a coral person and chime in.
We could zoom on it a little bit
Sure.
when we get the chance.
The one on the left,
Yeah, the one on the left.
zoanthids all over
Either
it.
that or zoanthids, yeah. I can't tell if it's a chrysogorgid or...
You're going to hit the, you know what to do, Mike. The other light.
The light.
Okay, you can zoom there, Marley.
Reminds me a lot of, I'm going to write down possible, although I'm not a great coral person. So. But this, Yeah.
Could be zoanthids on an old
Yeah,
black
it looks
coral.
like a different color. I don't know.
Zoanthids on a
Usually
black
they're
coral.
more gold, well, golden, although I know the gold is about the skeleton. All right, well, took pictures of it. Cool squat lobster.
Rough pictures there.
We can move
Bouncy,
on.
bouncy.
Whoa, aggressive.
Big though. Look at that.
Okay, so it's a zoanthid overgrowing an octocoral, apparently.
Yeah.
Thanks, Amanda.
Option B then.
Got excited. I know we had some of those on our list.
Yep.
Beautiful. Great shot.
Some prime real estate there.
Yeah.
Chirostylide, I forgot about that name, Amanda. That is crab. All right, Dan, time to get into a weird canyon and hand it off in a suboptimal condition.
Dust up some mud a bit.
Yeah, right.
Just
Make
like
it so they can't see.
Too cool up here though.
Yeah.
Freaking massive corals.
Yep.
Yep. Jenny's suggesting we paint it with the lasers if we see a big one.
So if we see big corals and happen to be zooming, we'd like to look at the laser as well. All right, got it. I'll pass that along.
Erica, do you think this might be a good spot for a Niskin?
Oh, good call.
Yeah.
Yeah, we've got Personella and some other things around.
Oh, why'd I do that? I didn't mean to do that.
But
And
I
then
think
Hey.
I think is there one or two left? I think on the last
There's 3.
There are 3, yeah,
shift.
we have 3 free.
Okay, I was listening on the last shift and
Yeah, one got
did they
double
pull
pulled.
2?
Yeah, that was numbers 2 and 3, so we still got 3 open.
Okay.
All
yeah,
right.
let's go ahead and do one here.
Cool. Would we like to do that real quick before shift change, ROV?
Yep, sure.
All right, let's sit down and we'll pull Niskin 4.
Roger.
They're primnoids, and what are the other main coral we're seeing around here so I log it in sample description accurately?
Paragorgia.
Paragorgia.
There's Gersimia.
I do not know how to spell the last one, if you would like to throw that in the chat.
Do that.
You on bubble or are you okay?
Oh, this thing's all jacked up because can you check the toolboxes? I might have opened one inadvertently. Yeah, they're all
looks
closed.
fine.
The starboard one might be open. Just bump it closed once. When we reboot it, messes all the
rocker
Oh,
stuff.
the Gersimia, the earlier one.
Okay. Yeah, it's closed.
Roger. You'll have to turn on the port shoal light.
Porch light is on.
Joe.
So you want number
No idea.
1, 2, 3, 4, which
Want the
is
orange one.
that is correct, the orange one.
Okay, we good to pull?
Yep.
There you go. Got it.
Just the one here?
Just the one, or correct? Amanda or Erica?
This is Meredith getting set up. Just the one will be fine.
All right.
Done.
Sorry, I missed that. Which Niskin number was that?
4.
4.
4.
Got it.
Number 4.
And that was sample 40.
40. 4 on 40. Got it.
Okay. Data going off comms to hand off.
Yep, handing over. Thanks the science team on shore. Appreciate
Thank you very much.
Yeah, thank you very much. That was just awesome.
Yeah, pleasure working with you guys again.
Fantastic dive.
Yes.
Thanks for the samples.
Good afternoon, everyone. They are doing a watch change right now. I am joining the watch change from shore, as is my compatriot Heather Coleman. This is Meredith Everett. I was on watch from 4 to 8 this morning, and I'm on watch again from 4 to 8 this afternoon.
Hi everyone, this is Heather Coleman. This is my first watch ever on Nautilus, so that's pretty exciting. I'm just glad I'm here with Meredith because she is very knowledgeable about all of these things, so I couldn't ask for a better co-host. Meredith, Lizzie Duncan just asked what you saw this morning from 4 to 8 a.m.
We saw some actually fantastic, enormous paragorgia, some of the biggest, nicest specimens I've ever seen. We also saw more lepidisis style bamboo corals. There was a lot of peristenella. We got into areas where there were probably 5 or 6 different coral species altogether. A lot of really nice big glass sponges. And so that was all. Those were all really fantastic to see. Poor Tom was going to get a little unlucky on his transects because at least for the first 2 watches this morning, it was like the rocks were spaced every 200 meters. We totally did not do it on purpose, but like every time we would start a transect, we would get right out of the rocks and into the sand.
Oh no, that's funny. Let's see if we can do better this time. Looks like Kurt has just volunteered to make me an inaugural watch certificate. Thank you, Kurt. Sounds great.
I'm working on getting on my computer right now one more window open, but I can see the nav screen as we're looking at the data here.
Abby Powell is putting in the chat all the great things that they sampled. I guess on the last watch, I had no idea that a sheepdog sponge is something you guys have been sampling, but that sounds amazing.
It's one of the kind of hairy barrel sponges, and the Midnight to 4 watch started referring to it as the sheepdog sponge, which I think is a great name.
From Midnight.
Kind of stuck for kind of stuck for the rest of the watch.
It's fantastic. You guys have the best names for sponges. Love it. Because it helps that they're not officially named yet. Call them whatever you like for now. looks like
Heather, on
Tom
that,
Leidig's offering. Go ahead.
I would say on that dive data recovery report, at least it looks like I got edit access now, so you may too if you have just added.
Okay. Not yet, but I'll keep refreshing. I think Tom Leidig's offered to set up some sort of cash prize for the first inaugural watch, whatever we're calling this. So I'm very interested to see what Tom Leidig and Kurt Whitmire have in store for me. Keeps getting better.
Hi, folks. This is Susie Cummings, just coming on as data logger. Nice to hear you all.
Nice to hear you too.
Were you on
Thank
the 4 to 8 this morning?
Yes, I was.
Okay, excellent. I'm liking the look of the patch we've landed in right now. I'm glad they took a nifty.
Mm-hmm. So we're sampling the water right now?
It was just sampled. It was the last thing the last watch did right before they started transferring over. So we've got
Excellent.
Susie on as our data logger, and I'm sure in a second, or probably not a second, but a couple of minutes, the pilots and navigator will finish their transfer and chime in as well.
Yeah, right now it seems like they're just talking over things, getting ready.
Yep. As every watch changes, each crew handing off to the previous team usually does a rundown of everything that went on, where they are in the dive, if there were any problems, just so everyone's prepared.
Mm-hmm.
And since we're the last watch of this dive, they may be talking about what they're going to do for recovery procedures as well.
Okay. Well, since we're waiting, Tom Leidig has mentioned the moldy Swiss cheese sponge, and that has piqued my interest so much that I just can't let it go. Meredith, what does the moldy Swiss cheese sponge look like? I mean, I can imagine, but
So
I'm sure there's more.
it is unfortunately not a sponge that we've ever seen this far north, but as we're going to be diving in sanctuaries down the coast, it has been seen from about Central California south into Channel Islands. I am sure we will encounter it during the Channel Islands leg. And it is a small demo sponge that grows kind of encrusting on the rocks. And it is kind of a dark blue-gray, and it really does look just like moldy Swiss cheese.
Love it.
We've only ever been able to sample one. And we'd love to get another because when it came up, it was a case of objects in the mirror are smaller than they appear. And what looked like this giant sponge on the video screen came up as like quarter size that we're now carefully like slicing little bits out for genetics and preserving for taxonomy because no one knows what it is.
Yeah. Excellent. Well, I hope we see another one on some of the later legs of the cruise then.
And it is in the master collection list. So if you've taken a look in there, if you look at the sponges tab, there should be a picture of it.
Okay.
And for those watching at home, it's not accessible to the public, but one thing we've done for this cruise and actually for the whole season is put together a list of coral and sponge and then actually other collection targets so that each watch and each science team that's working together knows the kind of thing we're looking for samples. And then these samples will be sent to the Museum of Comparative Zoology at Harvard, as well as to some other local labs. I take all the eDNA samples and process them so that they're available, and the museum samples are available to the community, and we use these samples to better understand these habitats.
Excellent. And so better understanding these habitats is a big part of the West Coast Deep Sea Coral Initiative, which we are starting to wrap up. It's almost the end of the government fiscal year, and we've had two fantastic years of funding this initiative at $1 million each, and that might sound like a lot, and it is a lot, but doing deep sea research costs quite a sum of money, considering the ecosystems that these animals live in, the high pressure, the cold temperatures, salty conditions.
Hello. Hi.
It's excessive.
Hello.
All right, I think we're settling in. Yeah? All settled? How's it going?
Yes.
All right, onshore team, so you're 4 to 8 here. Aaron, Jess, Kim in the front row. From what I understand, we were last kind of working our way into this little box canyon, doing just some exploratory work, but I hear there may be a transect in the near future.
It will be in probably the notes from on the nav screen, but we try to get 100 meters at least, between 100 and 200 between transects. So if we're already at that spacing, we could probably start with the transect just about right off the bat.
Okay. Yeah, you're 200 meters off the end of that last transect. The only concern is
Okay,
it's a
perfect.
bit hairy here if you want to go in and out of this canyon. The other option would be to traverse like just along a contour. That's a little easier to do and keep speed. But we'd have to probably get up around this feature, or I think we'll get into a gap here. I'm not quite sure.
Okay, why don't we, if you think it's easiest to get past the feature first, we can do that. And I'm happy to do the transect just traversing at the same depth contour. That would be just fine.
Okay, sounds good. So do you want to should we go up, kind of get a feel for the end of this box canyon that we're kind of in, and then maybe step up to where it is a little less crazy, and then kind of do our transect up there? Yeah, I think that sounds
Yeah,
good.
let's do that.
Okay, and then we'll kind of be making our way to 1.12. We'll probably end up a little north of it, but we can adjust then. Okay.
Yeah.
Yeah, sounds great. All right, so I'm thinking Argus is at 344. I think if we come to 355 and do some 20-meter steps, that will bring us into there. And then we're gonna probably just want to come up and go up, seeing depending on what we see.
Yeah, that sounds good. Yeah, we can. Yeah, because we're gonna aim for kind of that depth contour there, like a little
I'm thinking
bit.
like here.
Yeah.
I think that, so if you're watching back row, I'm thinking somewhere around here maybe we'll get kind of a ledge, maybe. We'll see.
Yeah. Okay. Let's do it.
All right. Bridge nav. Can we get a 20-meter step bearing 355? I'm guessing that purely off the sonar, so if it starts to look hairy to you, let me know.
Sure thing. And kind of the distance there will be around, what, 60 meters or so?
To up here?
Yeah.
40 to 60.
Okay.
Yeah.
Sounds groovy.
Yeah, we've got some nice NOAA corals that we're passing by. You get to see what those are in the eDNA as soon as we get those samples up. We started moving. Heather was talking about the West Coast Deep Sea Coral Initiative. And as part of this initiative, we'll be analyzing the transects that we'll be starting one shortly. That's one of the things the coral program is supporting, as well as genetic work on these corals and sponges and taxonomic work. And we'll even be hopefully using some of the images to feed into a deep sea ID guide that will be available for future researchers.
Indeed, and I hear we're also making some photo mosaics and 3D coral prints out. I'm not sure exactly what the the term is. They're so new. But those are some fun outreach products that are coming out of this initiative as well.
These are Paragorgia. These are the pink corals we're looking at here.
Bridge
These are
nav,
actually
another step, same bearing.
different species than we were seeing this morning. This morning we were seeing these very large, very dark pink, elaborate fans. They would be called usually Paragorgia arborea, but we have some evidence that they're actually a different species. So one was collected, and we have some colleagues doing taxonomy in that group. And then we have, these look like they're probably Paragorgia utlenics. They have this lighter color and morphology and are found at these depths. We actually have some of these maybe black corals, big chrysopathes that are mixed in here.
I'm seeing a fish, which I haven't seen for a while, and I'm wondering how many fish have you guys been seeing on these transects and exploratory sections of the dive?
On my watch early this morning, the only fish we were really seeing were deep sea sole and thorny heads, but that was very consistent with the depths we were operating at. We did also see some occasional grenadier, but you don't really see rockfish in large numbers or more of the flatfish till you get up closer to that 600-meter line.
Right. And what kind of sponge is this that we're just seeing in the middle of the screen?
This is one of the heterocony type sponges. You'll also hear them called goiter sponges. Two of these have been collected. We're not actually sure how many species there are of this group of sponge. The commonly known one is heterocony calyx, but it's based on old descriptions. This is a sponge that forms glass sponge reefs up in Canada. The skeletons, after they die, will actually fuse together and form these reef structures that other sponges will then continue to grow on. So Abby Powell, who just ended her watch, is very interested in doing the taxonomy with these sponges.
Great. And do they always have a squat lobster? I'm guessing that's what that is in the middle, and a crab on the outside, or who are they usually associated with?
So these occur across a range of depths, and you will see these
Bridge
with
nav,
crabs.
10-meter step, same bearing.
You'll see these with crabs, like that crab that's right next to it. In them, once you're shallower, you will see rockfish in them.
Maybe we should reset it. Are you okay for a reset? Oh, no, you're okay. It'll be close enough. Okay.
Meredith, it looks like Lizzie might be interested in 3D scanning that sponge, I think.
Wait, is that a stop?
I would actually I would recommend against that. They get really fragile when they're brought aboard, like they just crumble. And so it would be very hard to preserve that one for 3D scanning.
Got it. Thank you.
Now, if they did the photo mosaic and we can get some good structure from motion, we could probably get someone to build a 3D model off of that.
A 3D model from the photo mosaic. Have we tried that before?
I don't think so, but it's the new territory. Once I'm, right now, because of the current COVID situation, I don't have much access to my lab, but once we're back, we're going to be doing some scanning electron microscopy for doing coral taxonomy. But one thing, I found some papers on a technique to basically look at the sclerites, which are these little sand grains that the octocorals use for their skeletons. And you basically do photogrammetry by taking images with the electron microscope from all different angles, and then you can stitch those together and build a 3D model, which would, if it works, would hopefully allow us to print sclerites at full size rather than tiny little images. But it's something I haven't tried yet, and it would be interesting to play with.
Bridge nav, 10-meter step, same bearing.
Yeah, it's an exciting new area and some really great outreach products that are coming out of this work. So some of my favorite products from the West Coast Initiative. Since we're not seeing anything on the screen right now, I was wondering, maybe people are curious how our deep sea coral initiatives work. And if so, I'd be happy, since nobody can tell me not to describe them a little bit. Basically, we rotate around the country and have just spent the last 2 years focusing on the West Coast. What's up next is Alaska. So we have a ramp up year right now that'll soon transition into research, deep sea coral and sponge research in Alaska, and also data analysis of existing research that haven't been as thoroughly annotated yet as they can be. And that's all, of course, dependent on whether we're allowed to do field work based on COVID situation. But we'll have 2 years in Alaska, and then we'll move over to the Northeast US waters. That's an exciting place to be, since the New England Fishery Management Council and the Mid-Atlantic Fishery Management Council have both recently protected very, very large areas of their ocean. So we're excited to see more about what's in those areas. And after that, we'll go back to the Pacific Islands, where we were just researching deep sea corals and sponges, I think 3 years ago. We'll be back there, and then back to the Southeast US waters. So we rotate around the country more or less in that order. And we'll be sad to wrap up in the West Coast, but happy to spend some time in Alaska coming up soon.
Yeah, that's going to be a fun initiative. And one fun thing that we've been able to do is, for the last 2 years, Nautilus has been doing dives in the sanctuaries on the West Coast. And so we have a lot of video data sets that will be part of the initiative from here.
Yes.
And Jenny has just chimed in that Nautilus is headed to the Pacific Islands next. They've done a couple of seasons out there as well, as with the sanctuaries. So it'll be cool to see that as well.
Absolutely.
So Tom,
and the Okeanos Explorer will also be headed out there after they do a little bit of work in Alaskan waters. Seems like everybody's going out in that direction soon.
Tom Leidig has just asked online. One thought that he came up with was the white paragorges live in the O2 zone. We have reds below and a higher O2 level water, and we have red shallow and higher O2 water. It's possible. It's one of the things we're looking for, and we're starting to really try to look at what types of habitats, and the habitat includes things like the oxygen level these individual coral communities are associated with. And we definitely have seen in other areas changes in the coral faunal distribution that seems to correspond with oxygen. There is some modeling data that suggests that some deep sea corals may be more tolerant to low oxygen levels than we originally thought. You actually can end up with high diversity in low oxygen areas. But a lot of that's modeling data. And so one of the great things about doing these kind of surveys is we can start to verify those models.
Absolutely. And who's that little coral in the bottom? Or sponge?
I think that is another paragorgia, just a single small twig.
So little.
So just a logistical question for the science team on board. I know we had in the dive plan a on-deck time of 8 p.m. So I'm just curious what estimate we have then.
SPL right now.
I am listening on SPL, but I'm not hearing any ship chatter.
Yeah. Hi, this is Andrew. The current plan is still on deck at 8 p.m. local time.
Okay. So what time should we target for off bottom?
That's a question for the pilot, and there's currently conversations in the front of the van, so they're not they're not listening at the moment, but we can follow up with them in a second.
Perfect.
At 1,000 meters, it's going to take us about 50 minutes, maybe a little less to come up, so we'll probably be leaving the seafloor around 7 p.m. local.
Okay, thanks.
Yeah, sorry, we're just having a little chatter of here. There's been some issues with the Herc topside components that they're trying to work through as we move along. So if we're off SPL and you ask a question, I've asked Data to kind of get our attention, and we apologize. Yes.
Oh, no worries. The ROV safety is the first priority. As we're moving in that, we're currently exploring in this small kind of box canyon feature that we'd seen on some initial bathymetric data that some geologists were interested in as part of our transects and surveys. There's a small octopus up in the corner. And what's interesting here is this nice, like, ledgy, looks almost like carbonate-like rock that we're encountering. Got some yellow sponges on that rock. In some sponges, but not all, that yellow color comes from bacterial associates.
I didn't know that. Are there other sponge colors that come from bacteria?
You know, I don't think that's very well known. And I think looking at those kind of bacterial associates and symbionts in species like sponges that you don't necessarily associate with them is a rapidly expanding area of research.
On that subject, I know the bubblegum corals are pretty brightly colored. Is there a known reason why they're like so bright red and pink and those colors?
In those, it's actually the sclerites, so the little sand grains that give them their kind of skeletal structure. They're these like interlocking, elaborate grains of sand, basically, are actually those colors, bright red.
Wow.
And so in the ones we're seeing now that are kind of, they're of a lighter pink, but then if you were to zoom in on them, they have very, very dark pink polka dots every time there's a polyp or perculum. Those sclerites are actually two different colors.
Oh, cool.
And that's actually one of the characteristics we look at for taxonomy when we're looking at sclerites. We look at it both with an electron microscope, but then under a light microscope, because the color of the sclerites can be a characteristic.
Meredith, I would love your thoughts on something sponge related that I read recently. Was how sponges in some sense can be considered somewhat immortal, how their cells are much more simple than ours, and so they can kind of reorganize in a way. And it was just a little snippet that I read, but it was fascinating, and I'm wondering how accurate you would call that.
I mean, it's accurate in a fashion, kind of like the way they talk about some of the oldest and largest organisms being clonal colonies of aspen trees or fungi. So in some of these sponges, you do have these specialized cells that can basically become any other cell type of the sponge and can replicate in different ways. And so if they're reproducing clonally that way, you can have fragments of the same genetic lineage sponge that just live as long as those cells keep replicating.
And I read they could even go dormant for a little while and then reorganize into a bigger sponge again later when conditions are better. It's just incredible.
Yeah. They're one of these lineages of animals that we just don't know that much about. We know they're quite basal on the tree of life, but they just haven't received a lot of scientific attention until very recently. And so we're really interested in doing more work with them. Both because they're, here we're surveying in what's called an essential fish habitat zone. And we're interested in them as habitat, but at the same time, we're also interested in them for things like biopharmaceutical research. There was a cancer drug that was discovered in a sponge. And so that kind of thing is really of interest to us as well. So throughout the initiative, we've really been targeting the collection of sponges just to get a better handle on what's here.
Fascinating. Is that the little green sponge that I think was targeted for, was it pancreatic cancer?
Yep, that's the one.
Excellent. Here they're looking for that one in Alaska in particular.
Mm-hmm. I think
Awesome
actually
research.
we have that genus on our collection list for the coast as well.
Perfect. Speaking of sponges, there's another one.
Yep. And right next to it, there's a kind of a vague, frilly pink shape just to its right, and I think that is a little Garcinia coral.
All right, so we feel pretty comfortable starting transect from here if you guys are ready.
Okay, yeah, that would be great.
All right, so we're gonna be heading just to the generally about 060, but we might have to make some minor sways in and out on the way.
Sure thing.
Yeah,
I'll
works great.
I'll keep this heading here so we can see the wall and do a little bit of a, yeah.
And you're gonna just lateral along the wall?
Yeah, lateral along.
All right.
Is this view good for you guys, science?
Yep, that sounds perfect.
Okay.
All right, I'm gonna do 065, and I'm gonna start with 20 meter steps because I'm not as brave as running down.
Sounds good.
Bridge nav. Can we have a 20 meter step bearing 065?
Yeah, I'll fix that.
Thank you.
I don't know how double tapping this thing.
Hello, 4-8
All
watch
right.
team. I just want to let you guys know that we were going to open up the website to Nautilus Live viewers to ask questions. So I will be taking a look at those and asking each of you to join in.
Oh, Nicole's in the back row. I'm like, where is her voice coming from? Is she coming from the lounge? Or is she?
Yeah, I'm excited. We get to host our first Nautilus Live Q&A session for the cruise.
Amazing.
Excellent. I'm looking forward to it.
All right. Sorry.
We're just waiting for the ship to get moving and then Argus to start moving.
Yeah, the ship has a move in, which will start to pull Argus, and then Jess will start moving along with that. I'm putting the transect start here, but we can move it if we decide to wait until we pick up some speed. Is this number 6?
Oh, let me look at the log. I think it's transect number 9, 10. I think it's transect 10.
Oh, people have been busy. Great.
Wow.
Yep.
Bridge nav, another step, same bearing.
All right, I'm gonna start stepping.
Oh, there's a whole line of Garcinia coral right on that little edge there. It'll be lovely for Tom to count. Yep, Erica just confirmed in the science chat that it is transect number 10, so we're correct.
Okay, thank you.
So Meredith and Heather, you guys are leading this dive from shore. Do you want to talk to folks about what that's like? What's the experience of not being aboard the ship, but watching all of this and directing the ROVs like?
I have to say it's really different. You definitely are lacking the social camaraderie that you get on the ship, which is always a really fun experience. On the other hand, this morning I was able to run a dive in my pajamas with a large cup of coffee, which was nice. So that's I suppose a fun perk. And then there's different things like we don't think about so much, like when you're in the van, you've got a nice dark room to watch these huge monitors, and the noise level is pretty relatively controlled. There's not a lot of chatter. Here, it's like you have to remember to quickly mute if my dog comes running in and barking. So it's different, but it's fun, and I'm actually so happy that we were able to set this up in such a way that we can participate like this from shore. So it's fun to be so directly involved from my home office.
Yeah,
I
absolutely.
did use my
I've
Nautilus
actually never,
coffee mug.
I've never been on
Oh, go
Nautilus,
ahead,
so I
Heather.
don't have a Nautilus coffee mug, although I would definitely use it if I did have one, maybe one day. It is kind of fun to be sitting in the pajamas if you want to be and have, you know, all your computer screens on, but it is pretty isolated being, you know, alone and not being able to poke Meredith and be like, what is that, while we're on mute. You have to sound a little more intelligent individually, I guess. Harder to group think. One constant though is I have a bag of goldfish next to me. That's the same no matter whether I'm on a ship or shore.
Do you say you have a goldfish next to you?
Oh yeah.
You bring
Not a
him on
real
the
one,
ship?
the kind you eat. Yeah, absolutely.
That's delightful.
Meredith prefers the cheddar, which I have right now. I'm a big fan of the Parmesan, but you know, eat what you got.
I really thought you meant like a live goldfish, like in a bowl.
Next time.
With cheese.
My goodness.
Science on shore. Do you guys mind if I sorry, real quick, do you guys mind if I change my heading a little bit? I would prefer to kind of look a little bit more into the slope as opposed to kind of back down the slope. Does that make sense? So do you mind if I turn my heading a few degrees?
No, that would be just fine.
Okay, great. Thank you.
I just went over a bamboo coral there, right at the bottom of the screen. We're also getting some little Swiftia twigs and some Gersemia, which are a soft coral. They look just like the soft corals you'll see in the tropics, and they're fully inflated, and they all seem to be growing right on that little ledge there. They seem to have these little ledges that are providing hard habitat here.
So one of our viewers asked about the latency between when you see something on shore and when the command is executed.
That's much more comfortable.
And I kind of feel like for you guys on shore, you might not notice the latency as much as there's a latency between just the ship moving and the vehicles able to move along with them. I don't know if Erin wants to talk a little bit about what she's doing up there as we move along on this transect.
Yeah, so we're at nearly 1,000 meters depth, so it does take a while for ship movements to make it kind of down the cable and bring Argus along. So part of my job, which I'm not sure I'm doing really well right now, is to keep Argus away from walls, but looking at Jess as we move along. So Jess being Herc.
Oh, yeah, this is great.
So we use kind of a combination of a mesotech sonar that we're looking at and the bathymetry that we have to try and avoid any major obstacles or anything like that while keeping a nice steady speed for these transects.
A small field of feather stars here. These are crinoids. What's fun with them is occasionally one will come off the bottom and swim if we're lucky.
Oh, all good. Just making sure one of my two lines here parallels you.
One of the other different things about working from shore is when I've worked with Nautilus before, there's always been a direct communications person on board who's reading the Q&A from the public. So it's always nice to get those questions. So it's nice to have the opportunity to do that now, even though there's not a dedicated comms person sitting there telling us all in our ears, gotta keep talking, gotta keep talking.
Yeah, I admit I'm a poor fill-in for that, but people are sending some interesting questions in, and one of them is how many species of deep sea eels do we come across? It's an interesting one because I don't think there are many eel experts that we've worked with, but you guys on shore who've watched a lot more of this Pacific Northwest exploration, what do you guys think?
Yeah.
I think there's only a couple species that we encounter out here, although Tom and Erica have both just signed off, and they would know better than I would. But I've only seen a couple of like the deep sea conger type eels. I'm sure there are more, but I'm less familiar with them. I'm sure it's also a group like almost all deep sea groups that we just don't know how many species there are.
A sort of follow-up question, a viewer wants to know if you guys have a favorite plant or animal that you see on expeditions.
My favorite lately has been the big Primnoa pacifica corals. You'll hear it called the red tree coral in Alaska. It's a species that's very well known in Alaska and can actually occur quite shallow up there, but we've been seeing it all the way down the west coast, which was kind of a surprise for us. And they're quite big, quite striking. You'll usually see rockfish roasting in their branches. If we get to the right depths, we may see some tonight. They're my favorite of these expeditions right now. And then my second favorite is it's always nice to see a Dumbo octopus.
Nice. I didn't know we get to pick two. Well, I'm going to pick two as well. I think my favorite has to be Iridogorgia. And I'm guessing people may not know just offhand what that is, but if you can Google Iridogorgia, it starts with an I. I'm not going to spell the whole thing, but there isn't much else that's spelled like it, so you should come up with something. It's a magnificent spiral coral. At least I think most of them are spirally. And it's just so striking to see. There's nothing else like it. I know there's a lot of them in the Pacific Islands region. I don't know how many occur on the West Coast. It's a gorgeous, gorgeous coral. And for my second one, I've always been very fond of the sea hare, although I don't think those are deep sea creatures. I've only seen them shallow.
Stand by, Science on Shore. We got, we're pausing the transect here. We got a little bit of a topside crash again.
Okay, yep, and we can actually just pause and pick up again at the same spot.
Yeah, go ahead. We got controls back. Go ahead and try clicking it again. I don't think it took. Yeah, it didn't take. You want to go ahead and do it again there? And come up on the winch. Good. Great, thank you.
Science, once we get it sorted out, we can just restart the transect. We were about 60 meters in, but it's not a big deal.
Yep. That's standard practice for us. If nothing happens, just note it as a pause and then just pick it up right where we left off.
I think it's a continue. Is that correct?
Yes, yes.
Great. It's a continue, yeah.
A viewer wanted to know about those two green dots in the middle of the screen. Those are scaling lasers, so they're 10 centimeters apart, and they're really useful to be able to measure things and know the size of the things that we come across underwater, since it can be very deceptive depending on the zoom of the camera, the altitude of the vehicles.
Come back in front of Argus here real quick.
Yeah, on the quantitative transect, we've been running
That's actually how I noticed that it crashed.
The scientists from Southwest Fisheries Science Center will be analyzing video data. We'll be using these lasers because they count everything over 10 centimeters.
Let's see here.
Meredith, was that 10 centimeter rule?
Hey, Kim, you want to come down a little on the winch there? Trying to find out where we're at here. It seems like I'm getting tugged. So that means that I probably went.
Pacific Black Dragon.
was wondering if the count everything over 10 centimeters rule was instated after many hours of counting the tiny stuff.